Hi everybody, my name is George Huang. I'm going to be spending about 5 to 6 minutes talking to you about a project that's going on in my lab right now. So I work primarily with a bacterial pathogen called C. difficile. Some of you probably heard of it as C. diff. So I have another talk on our research day that where I'll talk more about C. difficile. Here I'll just really quickly go over what it does. So it's a human bacterial pathogen, and it infects people whose gut microbiota has been disrupted, usually through antibiotic use. And that's actually one of the reasons C. difficile is a big problem, not due to its symptoms of these diarrhea, and even in some rare cases, it lead to death. The major reason C. difficile is so difficult to treat is that after giving antibiotics, these patients usually have relapses because C. difficile can form spores that are even more resistant to antibiotics. These spores can persist both in the environment as well as inside the human host to cause problems again when they stop using antibiotics. And that's why one of the main goals in my lab is to identify these novel antimicrobial products that are different from the traditional antibiotics. So in this project, what we did is we looked through the literature. We found that there is a commercially available probiotic it's called Clostridium butyricum meyieri. So what we found is this strains of bacteria produce something called a bacteriocin. So which bacteriocin are these antimicrobial products that are made by bacteria, and they're usually against only closely related bacteria. And since Clostridium butyricum is closely related to C. difficile, we hypothesized perhaps this bacteriocin is going to be useful against C. diff as well. So we clone the gene from this probiotic strain into E. coli, allow it to overexpress the recombinant protein, we purify it, and we test it in our lab whether it is useful against C. difficile. Okay, so I'll go really quickly over some of the data we have. This just shows that we were able to successfully purify this bacteriocin. And this figure right here just to show that within 10 minutes, there was a significant decrease in the growth of C. difficile when it's incubated with a bacteriocin. And in 1 hour, we basically see minimal growth of C. difficile because this bactericidal activity of bacteriocin is very, very quick compared to vancomycin, which is the traditional treatment for C. difficile still right now. So it appeared to be even quicker, fast-acting compared to vancomycin. Okay, and this figure right here just to show that the bacteriocin doesn't have any toxic activity. It only works against Clostridium. Basically, it doesn't work against human. Okay. And here showing that the minimal inhibitory concentration of this bacteriocin was most effective against C. difficile, also work against other Clostridium, but it doesn't work against other bacteria at all, which is not surprising, but still good, because we really want to find a product that only targets C. difficile and leave all the other commensal bacteria in our gut alone. And here is just showing that different clinical isolates of C. difficile that we have in the lab, they are all equally susceptible to bacteriocin, and that's great. Okay, and so here in an animal model, what we did is we first infect animals with C. difficile bacteria, and then we treat it with our purified bacteriocin, and then we track over time the amount of viable C. difficile that still remains in the gut of the animal. We can see that within 1 hour there was a 99% drop, okay, in the level of viable C. difficile, and that trend was appear similar 6-hour post inoculation as well, and that's great. However, one of the problems with our product is that it's still a protein product. It's a recombinant protein, so it's time consuming, it's costly. We are really hoping this to be a project that can be applied and translational. So we really want to narrow the size of the bacteriocin down to just peptide level. So what we did is we predicted the three-dimensional structure of the mature portion of the bacteriocin, and that's also shown right here. Okay, it's about 80 something amino acids long. Now highlighted in red are these lysine residues, which are positively charged, and these are the residues that we believe are responsible for its antimicrobial activity. Okay, so what we're doing here is we're going to artificially synthesize oligopeptides that correspond to different portion, different location of this bacteriocin. Hopefully, we can identify a particular part that's involved in specific activity. Okay, and we have confidence it's probably somewhere around this region here. Okay, and second of all, we're going to mutate these residues to study whether they really are involved in the catalytic activity, and whether by adding more positive charge residue, is that going to increase the reactivity of the bacteriocin. And a different approach in this project is we're going to use a probiotic strain called E. coli Niso. Now, this E. coli Niso is non-toxigenic. In fact, it has been used in many studies to be a biotherapeutic, and it's safe for human consumption. So we are right now undergoing experiments where we're cloning the bacteriocin gene into E. coli. We hope this E. coli will express it and secrete the bacteriocin outside its environment. And so the goal is to have patients that would take our engineered probiotic strain. It goes into the gut where the probiotic are not secreted and kills off the C. difficile. Okay, and so that's a really quick summary of my studies, and I welcome any questions. Thank you.